Good afternoon, everyone. My name is Sharon Rounds, and I'm the Associate Dean for Translational Science at the Alpert Medical School and Principal Investigator of Advance-CTR, which is a NIGMS-funded research infrastructure center that is improving research infrastructure in institutions in the state of Rhode Island. We're very pleased to host a monthly Advance-CTR translational research seminar. And this year, our series has been distinguished clinical and translational research scientists, featured distinguished clinical and translational research scientists. And today we're very, very pleased to have with us Dr. Eric Morrow. Professor Morrow is the Menkoff Family Professor of Biology and Director of the Center for Translational Neuroscience at Brown University. He is a physician scientist and molecular neurologist who is focused on neurodevelopmental disorders. He was educated at MIT and Harvard and has been here at Brown, building a really wonderful center in translational neuroscience that I hope we'll learn a little bit more about. Today, he'll be presenting work on basic neuronal mechanisms and translational studies, which have the goal of improving treatments for neurogenetic disorders. So Eric, thank you very much once again for joining us today. We really appreciate it.
Thank you very much, Sharon. Can people hear me? Those who I can see, I guess, is who's Sharon. Give me a thumbs up. Okay, good.
You got it.
Good. Thank you very much. So I'd like to start by saying, I'm honored for the invitation. I'm grateful for the invitation and the opportunity to talk about the research which I've done at Brown and with colleagues at Brown and now in our Center for Translational Neuroscience. My career's work has been focused on childhood neurodevelopmental disorders and neurogenetic disorders. I've been at Brown now for around 13 to 14 years. So I'll tell you about a good amount of that work. Sharon just asked me to say a few words at the outset to discuss, I guess, biographically what has inspired me to do to be a physician scientist, to do translational and patient-oriented bench-to-bedside type research. And it's sort of a hard thing to summarize in a few sentences, but I think what I'll say is I feel very grateful and appreciative that I've had really wonderful mentors who've sort of inspired me from the very early part of my training, frankly, from high school. In undergrad and medical school along the way. And, features of those mentors that I think have been critical have been generosity of one kind or another. So, my path has been inspired by mentors who come before me that I'm very grateful for, and trying to pay that forward. Another aspect of it is I love the science. And so, we'll talk today about neuroscience. And there are a few things that I really love more than looking at the brain through a microscope. And so that's one thing that continues to inspire me, the complexity of the nervous system. Such a complicated organ system doing such phenomenally human processes, and how this organ, how the brain develops is really an inspiring problem, and it's been a fun challenge. The science itself has been a fun challenge. And then the last thing, I think, and perhaps the most important thing that has inspired me, and this has something I've experienced because I'm a physician, but I don't think it's, strictly speaking, available only to physicians, which is working directly with the families. So I've had the pleasure and honor of working with families who care for children or people, adults, with developmental disabilities. And, these families started, many of them, for me to be, patients or participants in our research, but over the years have become colleagues and friends, and, working with them and understanding what their life is about, and the fact that they've really garner hope from the work that we do is inspiring. So I don't know if those are the few words to what inspires me to be doing translational science. So let's get into some of the science here. I'd like to start with this slide. Today I'm going to talk about rare neurogenetic disorders. And this slide picks up from where I just was discussing working with families. So I've had the privilege of working on a rare neurogenetic disorder called Christensen syndrome. From the point of gene identification, this slide is now going on a conference that we hosted at Brown, which was the very first conference for families affected by Christensen syndrome. This mom here, who's the president of the Christensen Syndrome Family Association, Debbie Nash, emailed me in 2011, shortly after she got her son's diagnosis of Christensen syndrome, which ended a diagnostic odyssey of finding the diagnosis, and I've been in constant contact with Deb Nash since then, helped her form this family foundation, and we hosted this first meeting. I'll tell you about Christensen syndrome. It's an X-linked disorder that profoundly affects boys, who are shown here in their strollers. There's some neurotypical siblings in here, but I'd like to show this slide because you can get a sense. By the way, this is the foyer of our medical school, and you can get a sense of the positive feeling that they get from coming together and talking with each other and talking about research. So it's really been an honor for me to work with this group. By the way, the work was, this paper was done by an undergrad, David Stein. So we've had undergrad involvement and student involvement with the families, and they get to meet the families when we have these conferences every couple of years. So this month is Rare Disease Month. The last day of the month is Rare Disease Day. Rare genetic disorders affect a lot of people. So there are approximately 7,000 rare genetic diseases. People might know we have 22,000 genes in the genome. So that's a huge percentage of our genes which can cause a genetic syndrome. This is affecting 30 million people across the United States, 350 million people around the world. The vast majority of those people are affected from childhood. So today I'm going to talk about brain disorders. I'm going to mostly focus on autism and neurodevelopmental disorders. I understand this is a broadly translational science, translational medicine talk. And so one of the things I'd like to convey in this talk, perhaps in the context of the broader seminar series, is that brain disorders are, they're interesting and they're very challenging. And this is in part because the brain, I think, is a relatively less accessible organ system. assessing function of the brain is oftentimes based entirely on behavior, which can be a relatively indirect measure of where and what the organ is doing, tissue is doing at a given time. But it's also a really exciting time because the ethos of what I want to convey is that genetics and genomics has really brought a powerful tool, a powerful logic, a powerful approach to dissecting these complicated brain disorders. And then there are other methods that have come with it, neuroimaging, transgenic methodologies, and then these are actually human stem cells derived from, reprogrammed from blood of a patient with Christensen syndrome, so induced pluripotent stem cells. I want to tell you a little bit about our Center for Translational Neuroscience. So this is a center that is run collaboratively between the Carney Institute and the Division of Biology and Medicine, the med school. I direct this center, and it's also led by Judy Liu. I'm actually a psychiatrist, so I'm an MD PhD psychiatrist, and Judy is an MD PhD neurologist, which is I think actually exciting and unique. I don't know of too many centers around the country that are co-led by psychiatry and neurology together. Our motto is it's the same organ, even though we may end up focusing on a few different disorders that clinically may fall one way or the other. The logic that we follow in this center largely is a genetically inspired one, where the work starts with families, and many of those families have come to us because because they have a genetic mutation that causes a neurogenetic disorder. We take that mutation to the laboratory and conduct mechanistic studies. And I'll describe the various labs in our center. My lab is one, Judy's is a second. We study disease mechanism in animal models and in stem cell models, among other experimental models, with the idea of then gaining information that will help us with diagnosis, treatment targets, treatment strategies. And there are treatment strategies in development that include small molecule studies, metabolite supplement studies, molecular and gene therapy studies, among others that are ongoing, with the idea that the preclinical and the translational information can be brought back to families. So these are the lab is based, the center labs are largely based at the 3rd floor of Ship Street. We have actually many affiliate labs that are active collaborators, but those labs that we've been able to recruit to the center include my own, Judy Liu's lab, Alvin Huang, who also is an MD PhD neurologist who works in iPSCs in Alzheimer's disease, Greg Valdez, who works in synapses and aging and neuromuscular disease, and more recently, Sofia Lizarraga, who works in autism. So there's a very nice community of labs on the 3rd floor of Ship Street, and I think this is a great environment for trainees of all types, but those who are particularly interested in disease-relevant science, translational science. You get to see a range of how different scientists develop their careers in this space and conduct their research in this translational space. So the mission of the center is to advance knowledge of the pathogenesis of brain disease and to translate this knowledge to improved clinical outcomes for families who are affected by brain disease. So it's fairly broad, but it includes a good amount of basic science mixed with patient-oriented science. And We do bridge between campus, basic science research labs, and clinical research. This is something I've done as long as I've been at Brown. We have actually developed a CTN neurogenetics clinic that many of the investigators have begun to use, that I'll introduce you to the folks who are working in that include genetic counselors, research assistants, clinical project coordinators. The patient-oriented work is done in collaboration with colleagues across several departments. So the center is very interdepartmental and interdisciplinary, consistent with, the Brown ethos of interdisciplinary scholarship. So I would like to transition to talking a little bit about autism and neurodevelopmental disorders. And trainees that come to my lab or come to our center often find themselves reading these older texts, which are incredibly pertinent still. So here's Cajal. We have these Cajal's texts around the lab, around the microscopes. As I mentioned, one of the things that inspires me to really do this work is the beauty of the nervous system, which was first documented by Nobel laureate Cajal. And then this paper, which I recommend people who are interested in autism, you can find it on the internet. The PDF was around the internet, by Leo Kanner. This paper is now 80 years old, but still pertinent and accurate. Leo Kanner described 11 children who had a disorder of social communication and cognition and language that became the basis of the symptoms that he's described are still the symptoms of autism that are used clinically. It's a very, very valuable paper to read. So transitioning to autism, it's a childhood neurodevelopmental disorder that is increasingly prevalent, affecting as many as 1 in 60 to 70 children. Worldwide, it's probably felt to be around 1%. And the total costs to families and society are profound. For example, the economic burden, the total cost per year for children in the US, hard to measure these, but in the range of 10 to 60 billion. And medical expenditures for children who are affected by autism are a good deal higher than neurotypical children. So I want to talk a little bit about, I introduced the idea that disorders of the brain are fairly complicated, in some ways less accessible than some other medical diseases. Autism is a highly heterogeneous diagnosis. And over 10 years ago, I wrote this review with my mentor at Harvard Medical School, where we hypothesized within the larger spectrum of people affected or carrying the diagnosis of autism, there was going to be a subgroup that at the time we and others were calling lower functioning autism, who were profoundly affected, were oftentimes excluded from autism studies because of intellectual disability. They have more medical comorbidity, oftentimes with epilepsy, maybe nonverbal or minimally verbal. But we hypothesized that this cohort was going to represent a collection of rare neurogenetic disorders, rare, more highly penetrant mutations that may represent neurodevelopmental syndromes that were perhaps broader than autism alone, but within the autism spectrum. And that some of these could even indeed be monogenic. And I think this hypothesis was in process of being tested. by another exciting thing that happened at about that time, which is the reduction in cost of genome sequencing as a result of high throughput, short read sequencing, massively parallel high throughput sequencing, such that the cost of sequencing came down. And, you know, one of the things that I'd like to convey today, if people really want to think about this, is that it has become true, and it's now considered standard of care, that a child with a new diagnosis, a new neurodevelopmental disorder, intellectual disability, or autism, it's standard of care for them to get genome analysis, including an exome sequence. They start with microarrays, but will oftentimes move on to exome. And the different practice parameters from several different specialties have endorsed this as standard of care practice. And this has actually put neurodevelopmental disorders really at the forefront, perhaps with cancer, for genome analysis in terms of diagnosis and even potentially in the future, precision medicine and care. And as a result of initially researchers doing genome sequencing in patients with autism and related neurodevelopmental disorders, and as a result of now pediatricians worldwide doing their own exomes as a part of clinical care, it's become evident, particularly over the last decade, that within the autism spectrum, there are actually likely hundreds of distinct genetic causes. Now, this does not explain everybody on the autism spectrum, but depending on how you're defining autism, it can be somewhere between 20 and 40% of patients with autism will have a discoverable, highly penetrant genetic mutation. And so this article commented on our recent research at that time, and our advocacy for an approach of a focus on this group that we called then low-functioning autism, and more recently, a Lancet commission has defined this group of profoundly affected autism as a subgroup that they've coined profound autism. So profound autism is a group of children who meet the diagnostic criteria for autism spectrum and tend to be nonverbal or minimally verbal, have intellectual disability, and are likely to require care lifelong, this being profound autism. And in this article, long before the Lancet Commission, we advocated for studying the genetic causes in these individuals. More often, they'll have discernible genetic causes, and no longer excluding them from studies, but instead including them explicitly in studies because they haven't been studied in the past. And so this group here that we call lower functioning autism has now been recoined as profound autism. I'm happy to discuss this when there's time for questions. Okay, so that's a bit of an introduction. I guess it started a little bit about my career, about our center, which I think is a really exciting place for translational science, for trainees in that space, and more broadly on the genetics, the use of genetic analysis, genetic sequencing, and the diagnosis of autism and neurodevelopmental disorders. In the remaining part of the talk, I'd like to talk about two such neurogenetic disorders that we've been studying in my lab. Christensen syndrome, I've told you some about, and GPT2 deficiency. GPT2 deficiency is a neurogenetic disorder that we discovered in 2016 due to mutations in a nuclear encoded gene that is a mitochondrial enzyme, glutamate pyruvate transaminase 2, that has gotten my lab and a number of us in the center really excited about the study of mitochondria in the brain and in neurodevelopment. In Christensen syndrome, I would really like to exemplify how we go back and forth between working with the families and studying the phenotype and studying cellular mechanism. The gene or protein affected is an endosomal protein. So I'll also tell you about how by studying the condition, we've learned more about a unique mechanism of trafficking that we wouldn't know about if we didn't study the condition. So, you know, the idea is that you're studying diseases because, of course, we want to develop treatment for diseases, but in studying the diseases, they elucidate mechanisms at the cellular level in ways that are really quite powerful. And I'll also talk about how the gene that is affected actually can cause, with mutations, 3 different recognizable syndromes. So there's more to the story than just Christensen syndrome alone. The themes of what I'm going to say about the research in my lab that I've done with really wonderful colleagues is that families are collaborators in our research goals. And as I was saying from earlier on, the tools for understanding human biology and this inaccessible brain are increasingly powerful. We think that it's important to integrate across levels of analysis. So working with families, working with genomes and genome analysis, and then working with experimental systems such as induced pluripotent stem cells or rodent models, including mouse and CRISPR, has allowed us to bring back the rat and use it as a genetic model. And I'll show you some data there. Again, the logic of human genetics, the power of human genetics to dissect these complicated problems is particularly powerful. The synergy of bench to bedside. One thing that I would like to convey is that the mechanisms that we learn about when we study rare genetic disorders may be able to inform the disease process in apparently relatively more common brain diseases. And both of these conditions I'm going to tell you about have taught us something about neurodegeneration and likely will have relevance to neurodegeneration as well as to neurodevelopment. So the central question in my lab is what are the neurodevelopmental mechanisms that regulate the formation and maintenance of circuitry? The approach that we take is following families worldwide, and I'll show you that we have worldwide registries for these rare genetic disorders, and my work has always involved going to some degree around the world to find families where we may be able to find informative genetic mutations. And there's this long-standing tradition of that in human genetics. The phenotype that we study are disorders of cognitive development. My lab has had a focus on disorders that have abnormality in postnatal brain growth. So I'll point out to you why I think the biology of human postnatal brain growth is particularly relevant to these circuit disorders. They come with phenotypes such as postnatal microcephaly or postnatal macrocephaly, and autism very much is a condition where after birth there's abnormality in brain growth, and there's been a lot of study of overgrowth of brain in autism, as well as undergrowth in a range of neurodevelopmental disorders. We tend to study recessive mutations because those are loss of function and most easily modeled in the laboratory. So this is actually a slide that I like to show because it conveys just the profound growth of the human brain postnatally. Now, many people on this Zoom will perhaps know of primary microcephaly. So that's when a child is born and their head circumference or their brain size is profoundly smaller than a neurotypical child. You know, for example, that may be caused by genetic causes or viral things, you know, such as Zika, when we all followed Zika, we became aware of primary microcephalies. And the mechanisms of primary microcephalies usually impede neuronal proliferation, which occurs in human embryonically. So this is actually really one of the profound and interesting things about the human brain, is that this profound growth, which you can see here, is occurring largely by mechanisms that are not related to proliferation of cells, because neurogenesis is largely completed prior to birth. This brain growth actually is driven largely by neurodevelopmental processes that reflect circuit development. So the growth of axons, the growth of dendrites, the growth of synapses, the myelination of those circuit tracks. And so we follow genetic diseases where this growth is abnormal, where generally the head circumference at birth is normal, but then there's undergrowth of brain, such as postnatal microcephaly, or overgrowth of brain, such as would be the case with macrocephaly. So I want to tell you about 3 phenotypes that we see associated with mutations in this Christensen syndrome gene, this endosomal sodium proton exchanger 6. I'll tell you about that protein. I'll subsequently tell you about the cellular mechanisms that we're studying, but I'd like to start out with how much we learn about the brain and the developing brain and these conditions by working with the families. So Christensen syndrome is the profound X-linked male condition when there's loss of function of this protein on the X chromosome. It turns out that in the course of working on Christensen syndrome, we have worked with families, and we've discovered that a subset, not all females, but a subset of females who carry mutations that would cause Christensen syndrome in males, cause a female carrier syndrome, a specific syndrome in females that is quite frankly understudied. With X-linked intellectual disabilities, the fields tend to focus on the males who are most profoundly affected, but oftentimes there are female carrier syndromes, and I'll tell you about the female carriers. And then what I would like to tell you about is that we have discovered mild mutations in NHE6 that cause more mild developmental syndromes. And in fact, children with the diagnosis of autism who don't have the profound disabilities that's seen in Christensen syndrome, autism patients are turning up with more mild mutations in NHE6, which speaks to this genetic and phenotypic heterogeneity of autism. And I'll show you a little bit of work in each one of these realms. So first, the protein. So I got interested in the sodium proton exchangers from my work when I was a postdoctoral fellow and junior faculty at Harvard Medical School. So turns out that there are 2 related endosomal sodium proton exchangers, NHE6 and NHE9. We discovered mutations in NHE9 in children with autism and epilepsy in rare pedigrees in the Arabic Middle East. Turns out at about the same time, Peter Strom, a pediatric neurologist, defined very similar mutations, loss of function mutations, in the X-linked form of endosomal sodium proton exchanger NHE6. This X-linked syndrome, which we subsequently called Christensen syndrome after Arnold Christensen, the pediatrician who defined the first families clinically, are more common. It turns out Christensen syndrome is probably among the more common X-linked developmental brain disorders, affecting 1 to 2% of the X-linked, 1 to 2% of the X-linked developmental brain disorders are mutations in NHE6, or the gene is also called SLC9E6. So that's about, Fragile X is the most common X-linked developmental brain disorder, which is about 5%. So this is in the order of about half as frequent, a little less than Fragile X syndrome. And we estimate it's 1 in 20 to 1 in 80,000 live births. These are pedigrees, which we first defined, again, through privilege of working with the families. This work was done by Matthew Pesco-Solido and David Stein. David was an undergrad who took my genetics class and stayed in the lab. And Matt, at the time, was a research assistant who joined the NSGP, the neuroscience program. And pedigrees are typically X-linked, where you have a carrier mother who transmits to an affected son. The females are carriers, and I'll tell you about the syndrome related to being a female carrier. It turns out that Matt and David were able to show in this paper that about half of the families are actually de novo mutations. So a new mutation on the X chromosome, usually that arises in the egg, but is not a mutation in the mother. And this is actually a mechanism that is increasingly discoverable and defined with exome sequencing. This pedigree down here is fun to show because you can actually see the mutation happen de novo. The mom is a carrier, but her biological parents, neither of them are carriers. So there was a spontaneous mutation on one of the X chromosomes that was transmitted to the mom, who then subsequently transmitted it to the son. The protein is a 12 membrane spanning protein. It sits in the endosome. It lets protons out in exchange for sodium. What I'll tell people, remind people, is that the endosome is a bubble. Things get endocytosed from the cell, receptors, receptor signaling complexes, and the endosome has choices. It can mature, it can recycle receptors, it can signal from within the endosome, or it can mature to the lysosome. And as the endosome matures to the lysosome, the pH within the endosome gets driven down, so it becomes more acidic. And we actually hypothesized that our exchanger functions in a realm of delaying the acidification of the endosome, and actually in this way affects maturation of the endosome to the lysosome. The mutations in Christensen syndrome are most often complete loss of function, and we've been able to show that these nonsense mutations are not truncating mutations, but actually lead to nonsense-mediated mRNA decay. We've shown that in the iPSCs from the patients. So the protein is gone in the patient cells. There are other loss of function like mutations, splice mutations. Occasionally we come across these missense mutations, which are probably more complicated than the pure loss of function. So this is our clinical team from the CTN Neurogenetics Clinic. This team involves a number of physicians, scientists, clinician scientists, researchers, and it is also led by a support group. Carrie Best is a project coordinator, longstanding, worked with families with neurodevelopmental disorders. Danielle St. Pierre is an experimental psychologist who works with the families. And then formerly an undergrad, again in my genetics class, Jennifer Elacio, who's now a post-bac research assistant. So we actually lead the Christensen syndrome natural history study. We follow patients around the world who communicate with us via Zoom. Occasionally will come and visit us. We now have the largest, it's actually the central cohort of families with Christensen syndrome with many states across the US. We're not doing as well on the West Coast. 4 continents, 15 countries, 13 languages spoken. So this human genetics is very much an international study. This shows actually age of the affected individuals with Christensen syndrome and the number of participants. Arnold Christensen defined that there may well be premature mortality, and unfortunately, this is what we're finding. And we've begun to understand better the causes of death and the progression of the illness across the lifespan into adulthood. We've also opened a brain bank, and families have begun to donate brains when their loved one passes away. So really, we are studying the natural history of this disorder across the lifespan, identifying the most relevant treatment targets for ideally interventions and clinical trials. The syndrome, all of the men, boys are nonverbal. They all have intellectual disability. They all have epilepsy. There's a cerebellar degeneration, which I'll show you, that leads to ataxia, progressive ataxia, and they have this postnatal microcephaly. About a third of them gain the diagnosis of autism. Many of them are diagnosed as Angelman syndrome preceding that, and there are a range of different medical-related conditions. This is just to show we collect the MRIs from the patients, again, from around the world. And many of these patients you'll see have had multiple MRIs, and we reconstruct these and draw computer automated morphometry. This is just showing the frontal occipital diameter. And you can see that the frontal occipital diameter consistent with the reduced head circumference is reduced. This is actually a patient I'll tell you about in a moment who has a hypomorphic mutation and is diagnosed with autism, but not Christensen syndrome. One of the themes of this work, which I'll tell you about, is that we conceive of Christensen syndrome as a mixed neurodevelopmental neurodegenerative disorder. So these are MRIs from the same boy at 4 years and at 10 years, and you can see there's a frank cerebellar degeneration that causes ataxia, and then there is, unfortunately, a progressive, although more slowly progressive, more diffuse degeneration across various different regions of the brain. And I'll tell you a little bit about what we've learned about the progressive neurodegenerative aspects in Christensen syndrome that's quite important with, I think, broad relevance to neurodegenerative disorders. And I'll tell you about that in a moment. So the female carriers, so I want to pause for a moment and talk about the female carriers. So for a good period of time early on, we didn't focus on the female carriers. And then it became clear to us that both developmentally and with aging, there could well be an associated phenotype to being a carrier of the CS mutation. Now, Christensen syndrome is on the X chromosome, and the way this works in the female brain is due to X chromosome inactivation, the brain becomes a mosaic of cells, either expressing the healthy copy of the gene or the mutated copy of the gene. And depending on if there's some skewing of X chromosome inactivation, females may be unaffected or more mildly affected or more profoundly affected. So we do see a good proportion of females who actually have no clinical diagnoses and potentially no major impairments. We see a good number of females who either developmentally or into adulthood have learning differences, ADHD, mild or moderate intellectual disabilities, even autism, and occasionally we see seizures, which we see 100% of the time in the boys with Christensen syndrome. We've seen psychiatric diagnoses, and then one of the areas that we're following very, very carefully is that we may be seeing an increased incidence of neurodegenerative conditions in females who are carriers. This is a typical presentation of a pedigree that we see where the family's identified because they have a boy with Christensen syndrome. It's discerned that the mother is a carrier. This particular mother was neurotypical in her development, had mild learning difference, but did not impede her ability to adapt and function in life. Her IQ here shows 100, which is, an average intelligence. And then she reported to us that her mother, who was a carrier of the mutation, got diagnosed with cortical basal degeneration, which is a neurodegenerative disorder that often has tau deposition. And then this is the MRI of her brain, and you can see atrophy here in the cortical regions. And so we've seen a frequency of this. It's still a small study, so we don't have statistical power yet to be sure that there's this increased incidence, but we're concerned that being a carrier could lead to an earlier neurodegenerative condition. We see with some frequency at this point atypical Parkinsonism as a diagnosis in the 40s and then the 50s. And then let me tell you about another aspect of the phenotype. So we see Christensen syndrome in those with loss of function males. We're characterizing the female carrier syndrome. This work, which is done with a psychologist, Julia Riggi. The female carrier work here has been done by Brian Kavanaugh. Brian's face was behind my Zoom screen. But when I saw Julia, I thought that Brian was in the prior. Brian is a neuropsychologist, developmental neuropsychologist at Brown, and Julia is a psychologist at Bradley. So we've seen a good number at this point of children with a history like this, where mom calls us or dad calls us and says, my son got exome sequenced, he has autism, and they found a mutation in SLC9A6 or NHE6. I read your papers. He doesn't have Christensen syndrome, but he's been diagnosed with autism. And so this is actually one of these children, a 10-year-old boy who's in an integrated classroom, who doesn't have ongoing epilepsy diagnosis, isn't on anti-epileptic meds, has had one seizure. So again, 100% of boys with Christensen syndrome will have epilepsy. So and epilepsy occurs in autism with about a frequency of 20 to 30%. Abnormal eye movements, which we actually see in Christensen syndrome, some awkwardness of gait, and then the IQ is in the range of a mild to moderate intellectual disability, and they meet criteria for autism, but the boy has language, and none of the boys with Christensen syndrome have language. This is the mutation that we find in this child, in this boy. He has a 5 base pair deletion just upstream of the splice acceptor site in an exon that causes abnormal splicing at a frequency that we've been characterizing in the iPSCs. So at some percentage of time, there's abnormal splicing and abnormal production of NHE6, but it's not 100%. So this looks like a hypomorphic mutation that's occurring in autism, and we're in the process of collecting quite a number of these cases. And this is showing you that boy has some mild cerebellar atrophy, which may contribute to his awkwardness of gait, which is not typical MRI finding for a person with autism. All right, so that's a little bit on the clinical syndrome. So I'm going to tell you quickly about some of the cellular mechanisms that we've been working on. It's actually quite interesting. The study of Christensen syndrome has taught us a good amount about endolysosomal trafficking. The protein sits in the endosome and lets protons out as the endosome matures. The vacuolar ATPase drives the pH down. Now, without NHE6, you get an overacidified endosome. And that was our hypothesis. And we and other groups have been able to show this recurrently. We showed it initially in neurons, and it's been reproduced widely by a variety of different methods and now a variety of different experimental contexts. This is work that was initially led by Sofia Lizarraga, who was at that time an investigator in MCB at Brown. She subsequently left, went to UCSF, started her own lab, and we just recruited her back to run her own lab in our center. And Qingyu Wang, who's an assistant professor in the center. These are mouse neurons from the knockout, and you can see in the absence of NHE6, there's a decrease in the pH. So it is more acidic within the endosomes. if you look at patients, these are neurons from patients with Christensen syndrome or their unaffected brother, and you measure the pH, you see equivalently an overacidification of the endosome. And then work that's been done by Li Ma, who was an investigator in my lab and in our center. She went into iPSCs from patients and corrected the mutation using CRISPR-Cas9, recovered the protein here shown on Western blot, and you can see a recovery of the pH in those corrected iPSC lines. Further work in iPSCs speaks to the potential of a gene therapy approach. So Christensen syndrome neurons tend to be less arborized. However, using gene replacement strategies, we were able to put NHE6 back into these neurons and recover the arborization. And this experiment was done by Abby McGuire with Sophia and Li Ma. And Abby earned her PhD from Brown a few years ago. And so I'll tell you one last part of the Christensen syndrome story, which we thought was paradoxical, but I think we've worked it out. At the same time that we were showing that the endosome was overacidified, a separate group, Steve Walkley and Peter Strom, Walkley is a lysosome disease neuropathologist, was showing that there was accumulation of cellular debris, breakdown products, in this case GM2 ganglioside, that would accumulate in disorders where there's lysosome deficiency. So we had this puzzle of how can you have an overacidified endosome but lysosome deficiency? It seemed paradoxical. And this was something that Matt Pescosolito worked out in his PhD. Matt did show that in Christensen syndrome neurons, there's a decrease in the degradative capacity of the neurons, shown here by endocytosis and digestion of a fluorescent BSA. And a postdoc in the lab who made a Christensen syndrome rat, I'll tell you more about that in a moment, indeed showed that GM2 builds up in the rat brain. So we have the mouse brain and the rat brain showing evidence of lysosome deficiency. Matt is able to show that in vitro. And then Matt did a series of experiments. I don't have time to go into all of them here today, where he basically showed that what happens is there's an overacidification of the endosome. This actually leads to precocious activation of lysosomal proteases in the late endosome, a decrease in fusion and delivery of healthy lysosomal endosomes to the lysosome. So you actually end up with lysosome deficiency, and a increase in exocytosis and release of microvesicles and exosomes from the neurons. And Matt actually showed this. He tagged the MVBs, the exosomes, with a fluorophore that fluoresces upon exocytosis. This is shown here. You can see he scores these microscopic exocytosis events in neurons using TIRF, total internal reflective fluorescence, and is able to show that there was an enhanced release of these exosomes in the Christensen syndrome neurons. And then just to conclude, the Christensen syndrome pathophysiology, getting back to the postmortem studies. So in the postmortem, other groups have shown that there was diffuse tau deposition in Christensen syndrome. This was not inconsistent with our findings of cortical basal degeneration in the female carriers. CBD is oftentimes construed as a tauopathy, particularly with both white matter, glial and cortical neuronal tau. And this is what was seen in the Christensen syndrome neuropath events. We made the rat because rats are actually much better models of some of the Alzheimer's related protein aggregations, such as tauopathies. Mice don't get tauopathies. It turns out that rats do. So we made the Christensen syndrome rat, and Eugene Lee, in collaboration with Tracy Young-Pierce, was able to show in these aging rats with Christensen syndrome that you do get both neuronal and glial tau depositions. And so just to conclude this Christensen syndrome part, NHE6's mutations are more than just Christensen syndrome. There's an important female carrier syndrome. There's also hypomorphic mutations associated with autism. It's taught us about the process of endosomal maturation. We conceive of the disorder as both neurodevelopmental and neurodegenerative, and it's potentially within the spectrum of lysosomal disorders and even tauopathies. So in the last minute or two, I'm going to talk about a different genetic syndrome that we worked on, just to give you a taste of this work in our lab. And this work was led by Qingyu Wang and Ozan Boitis, who's a graduate student. This is a mitochondrial disorder, recessive mitochondrial disorder that we mapped in large international pedigrees with recent shared ancestry. We identified loss of function mutations in the mitochondrial enzyme glutamate pyruvate transaminase 2. This is a disorder, again, following the phenotype is an incredibly powerful way of learning about the brain and doing science. The kids have postnatal microcephaly and intellectual disability, which is what we initially got interested in, but they also have a progressive spastic paraplegia, which is related to more common motor disorders, such as primary spastic paraplegia or even ALS. The enzyme moves an amino group from glutamate to pyruvate and generates alanine and alpha-ketoglutarate. And Ozan in his thesis was able to show that this enzyme plays a critical role in the production of alanine for protein synthesis in neurons and alpha-ketoglutarate for Krebs cycle function. We did a series of experiments, metabolomics in the brain of the knockout mouse, and I'll just show you this quickly. The PCA, the principal components analysis, shows separation of the metabolomics with the postnatal brain growth, consistent with the idea that this is an enzyme that nourishes the growth of the postnatal brain, and consistent with much of the work that Ozan followed up. Is there deficiencies in protein biosynthesis and Krebs cycle, among others. The reason why I point this out is metabolism disorders offer us the possibility of intervention in neurodevelopmental disorders in a really special way, and PKU is clearly the flagship disorder for this, where identified with newborn screening and modification in the diet, you can prevent a developmental disability. And this is really what we're hoping will be the case of GPT-2 deficiency. And in our center, we have a strong interest in neurometabolism because we hope that there are other disorders that can be identified like this, where with newborn screening, we can potentially prevent the development of intellectual disability. So GPT-2 disease, just to summarize, the second disorder is a neurodevelopmental disorder, again, with neurodegenerative features, playing an important role in neurometabolism, with the idea that maybe through metabolite supplements, we can treat this disorder. So I'm going to conclude. I thank the families who participate in our research. This is actually a Christensen syndrome conference just before COVID that was in the Netherlands. The folks are in here. Debbie Nash is in here somewhere. I'm in here somewhere. I got to go to that meeting. And then we just had the reunion, the not quite 10-year reunion in Providence in October of the original Christensen syndrome. And some of the original people affected who came to that first conference came to this one. With that, I will conclude. I'm grateful to many people and to many of the funders, including internal funding at Brown. I'm open for questions if there's still time.
Fantastic. Thank you very much, Eric. I really appreciate it. We are a bit short on time. I will ask you one question, however. Does this mutation of the sodium hydrogen exchanger, is it manifest in other cells besides neurons?
Yeah.
And, and are there systemic effects?
Yeah, it's a great question. So, and we see this a lot in neurogenetics, where there's a protein that is mutated, a gene that's mutated, and it's expressed in any or all cells of the body. So NHE6 is expressed in every single cell in the body.
Right.
It is highly expressed in the brain. And I don't want to say that there are not systemic effects, non-brain effects. Some folks have been studying it in bone. But by and large, the families are not presenting with primary non-brain problems that hit their top 5 list, if you know what I mean. I mean, their problems are seizures, motor. The children do, as they grow older, and we have paper in process to show this, become leaner and leaner. Their body mass index really goes down, and it's harder for them to gain weight. So there may be a metabolic sequelae, but we see that in neurodegenerative disorders. So again, it could be secondary to the progressive brain effects.
Wow. So fascinating work. Unfortunately, we're a bit over. So I do want to thank you once again and congratulate you on a really beautiful body of work. It's really impressive, and thank you again.
Thank you for the invitation.
Bye-bye, everybody.